is called growth. Then growth is accompanied by metabolic activities. It includes both anabolic and catabolic process. Particularly, catabolic process means respiration. This respiration provides energy. In presence of energy, cell division takes place, and with the help of cell division, that a growth occurs. Then, increase in length of leaf is a growth. But when wood is placed in water, it swells, but it is not a growth, because it swells due to absorption of water, not by cell division. So it is not a growth. And why that wood swells means it is due to that imbibition. It is growth. Next, growth is indeterminate. Here, unlike other organisms, plant growth takes place throughout life. So plant growth is called unique, because throughout life growth takes place. Why throughout life growth takes place means it is because of meristems. Meristems means immature cells. Always they undergo a division, and they present in different parts of the plant. They undergo division. Because of division, new cells are formed. Because of division, new cells are formed. These cells lost their power of division and make the plant body. So point is, in the plants, meristems present at different locations, and they add wherever that a new cells required. There that a new cells are formed. This form of growth is called open form of growth. Then next one, particularly meristems help in cell division. So meristems help in growth. Then types of meristems. Here based on position, meristems divided into 3 types. First one, apical meristems. Apical meristems present at the apex of root, shoot and branches. They help in linear growth. Then next one, intercalary meristems. Intercalary meristems present in between permanent tissues, and particularly intercalary meristems present in grasses like monocots. Then these apical and intercalary meristems, these are primary meristems. They help in linear growth. Then in some plants like dicots and gymnosperms, along with linear, this thickness or width is also increased. This thickness or width is increased by the activity of lateral meristems. Generally, lateral meristems present in dicots and gymnosperms, and this lateral meristems, they increases thickness or girth by secondary growth. Example for lateral meristems, vascular cambium and cork cambium. Vascular and cork cambium. Your point is total cork cambium is secondary, but vascular cambium partly primary, that is intrafascicular cambium, and a partly secondary, that is interfascicular cambium. So point is lateral meristems not only secondary, they are both primary and secondary, and not only secondary, even primary meristems participate in secondary growth. For that example is intrafascicular cambium. This intrafascicular cambium is primary meristem, participate in secondary growth and increases thickness. Next one, growth is measurable. Growth is measurable at cellular level. Growth is measured by increase in amount of protoplasm, but by seeing this protoplasm, measurement of growth is difficult. So there are different parameters to measure that growth. For this, one is increase in dry weight, then increase in fresh weight, then number of cells, then length, then volume of cell. So these are different parameters to measure growth, like increase in dry weight, fresh weight, number of cells, length, and also that volume of cells. For example, one 𝗧 apical meristem. 𝗧 apical meristem produce 17,500 daughter cells in 1 hour. In watermelon, 3,50,000. That fresh weight is increased. So point is, previous one, 𝗧 apical meristem produce 17,500 cells. So it is increase in growth by dividing that number of cells. Second one for watermelon, it is that volume of a cell. Even growth of pollen tube. Growth of pollen tube is also measurable in count of that length. And dicot leaf, increase in dicot leaf is also measurable by what means? By surface area. So in that way, growth is measurable. Phases of growth. Generally, root apex and a shoot apex, they shows 3 phases. First one, meristematic zone. So meristematic zone means it contains meristems. So these meristems, they undergo division. And the characters of meristems, these meristems closely arranged without intercellular spaces. They contains dense protoplasm and a large nucleus. and adjacent cells connected by that plasmodesmata. Generally cell walls thin and made up of cellulose. Then proximal to meristematic elongation zone. Proximal to meristem means above the meristem. There is elongation zone. In elongation zone, vacuolization, increase in vacuolization takes place. Cell enlargement takes place. Increase in vacuolization, cell enlargement takes place, and new wall materials are deposited. Proximal to elongation, maturation zone. In maturation zone, that cells attain maximum size. So in maturation zone, cells attain maximum size, then increase in thickness, and also protoplasm modifications takes place. Protoplasm modification takes place. These are phases of growth. Next, growth rate. What is growth rate means? Increase in growth. Increase in growth per unit time is called growth rate. Then growth rate can be expressed in mathematically, and this growth rate can be expressed by arithmetically and also geometrically. So first one, arithmetic growth. In arithmetic growth, parent cell divides into two daughter cells. Then among two daughter cells, one daughter cell undergo differentiation, then other cell undergo division. So arithmetic, parent cell gives two daughter cells. Among two, one daughter cell undergoes differentiation, other daughter cell undergoes division. It is arithmetic growth. Then arithmetic growth exemplified for what means length of root. It is exemplified for length of root. Then when graph is drawn between that length of root against time, that linear line can be obtained. Then arithmetic growth can be expressed by following formula: LT equal to L0 plus RT. Here LT means growth at time. L0 growth at time 0. Then R is a growth. R is growth rate. So it is for arithmetic growth. Second one, geometrical growth. Here geometrical growth means parent cell divides into two daughter cells. Parent cell divides into two daughter cells. Both daughter cells undergo division. Both the daughter cells undergo division. So, right, it is called geometrical growth. It means one parent cell gives 2 daughter cells, then 2 gives 4, then 4 cells gives 8. Geometrical growth can be exemplified for what means increase in fresh weight or dry weight or even number of cells. When graph is drawn between increase in fresh weight or a dry weight or a number of cells, that a sigmoid curve can be obtained. Sigmoid or S-shape curve can be obtained. Why means initial stage growth is slow. Initial stage slow growth. Why means here meristems present. Meristems undergo division. It is called lag phase. Then next, rapid growth takes place because here cell elongation takes place. That rapid growth is called log phase, or it is called exponential stage. Finally, at the top, growth is constant because it is senescent stage. So, because of this sigmoid curve or S-shaped curve can be obtained. For this geometrical means increase in dry weight or fresh weight can be obtained by a formula. That formula W1 equal to W0 e to the power of RT. Point W1 is a final stage. W0 initial stage. E base of logarithms E, then R, that is growth rate. Next, absolute growth rate, relative growth rate. In simple terms, absolute growth rate means final growth minus initial growth. For example, here there is one leaf. Its initial growth 5 centimeters. Final growth 10 centimeters. Here another leaf. Initial growth 50 centimeters. Final growth 55 centimeters. So, absolute growth rate, leaf A, leaf B. So, absolute growth rate for leaf A, final growth is 10, initial growth is 5. So, 10 minus 5, 5 centimeters. For leaf B, final growth is 55, initial growth is 50. So, final growth is 55, initial growth 50, So 5 centimeters. So in both leaves, absolute growth rate is same, 5 centimeters. What is relative growth rate? Relative growth rate means final growth minus initial growth by initial growth into 100. So relative growth rate for leaf A, 5, initial growth 5 by 5 into 100. So relative growth rate 100. For leaf B, final growth minus initial growth means 5 by initial growth 50 into 100 means 10 centimeters. So your point is, for both leaves, absolute growth rate is same, but relative growth rate, here 5 to 10 centimeters increased. So in leaf A, relative growth is 100%. Here, leaf B, relative growth, absolute growth rate is 10 because it increased from 50 to 55, 10. Next one, conditions. Conditions necessary for growth. What conditions necessary for growth means? Water, oxygen, even temperature and minerals. Water is essential for growth because water maintains turgidity. So water is essential for growth. It maintains turgidity, and only turgid cells in that cell wall shows expansion. The next one, oxygen. Oxygen is essential for respiration. So this respiration gives energy, and in presence of energy, that cell division takes place. In presence of energy, cell division takes place. Even for growth, optimum temperature is required because most of the enzymes are active at optimum temperature. Then minerals, both macro and micro elements. Both macro and micro elements essential for growth because these macro and micro elements, they produce that protoplasm. Along with this, even light and gravity also essential for that phases of growth. Next, differentiation, dedifferentiation and redifferentiation. Redifferentiation. Point. Differentiation produced from differentiation is apical meristems of root and stem. Phenomenon involved differentiation. Differentiation. In the formation of primary tissues like parenchyma, collenchyma, sclerenchyma, primary xylem, and primary phloem. So first one, differentiation produced from apical meristems of root and stem. Apical meristems of root and stem. And example for primary, even intrafascicular. Example is intrafascicular cambium. Phenomenon involved differentiation. Cells differentiate to perform different functions. This is differentiation. Next one, dedifferentiation. Dedifferentiation means they produce from permanent tissues, medullary rays, undergoes dedifferentiation, produce interfascicular cambium in stem, dicot stem. Then primary cortex by dedifferentiation produce core-cambium or phyllogen in dicot stem. Then in a dicot root, conjunctive tissue and a pericycle. Dicot root, conjunctive tissue and pericycle by dedifferentiation produce both core-cambium and also vascular cambium, in which means in dicot root. These are differentiation, dedifferentiation. Third one, redifferentiation. Redifferentiation, secondary tissues or secondary meristems by redifferentiation gives secondary tissues. For example, vascular cambium. For example, vascular cambium by redifferentiation produce vascular tissues like secondary xylem and phloem. Then core-cambium by redifferentiation it produce cork and also secondary cortex. Plant shows different adaptations for that environmental. It's called plasticity. So plant shows different developmental stages due to that environment. It is called plasticity. Example for plasticity, heterophyly. Heterophyly means presence of two different kinds of leaves on a same plant is called heterophyly. Example for heterophyly, coriander, scientific name Coriandrum sativum, then ranunculus, buttercup, then next one delphinium, that larkspur. So coriander, Coriandrum sativum. These are examples for heterophyly. Here hydrophyte, ranunculus, In ranunculus, rightly it is called environmental heterophyly. Ranunculus, it is called environmental heterophyly because ranunculus is hydrophyte, in which upper leaves normal perform the function of photosynthesis and submerged leaves are dissected and help in absorption of water. Finally, this growth development requires both external and also internal factors. External factors includes light, temperature, oxygen and water. These are called external factors are called extrinsic factors. Second one, internal factors, also called intrinsic factors or intracellular factors. These are rightly growth factors, intracellular factors, which includes what means chemicals or that phytohormones, plant hormones. So actually, also previously called chemical messengers. Presence of chemical messengers, first reported by Sachs. For these chemical messengers, hormone term was given by Starling. Hormone term was given by Starling. What is phytohormone means? Actually, phytohormones means produced in one place, migrated to other place, where they shows effects like physiological effects called phytohormones or plant hormones. These phytohormones, they promotes growth, so that is why called growth regulators. Some phytohormones, they promotes growth, so called growth regulators. These growth regulators, auxins, gibberellins and even cytokinins. These are growth regulators. But some chemicals, they inhibits growth, so rightly called growth inhibitors. Growth inhibitor, abscisic acid, ABA. But there is another hormone, ethylene. Another hormone, ethylene. Ethylene comes under both. It shows promoting characters and also that inhibitory effects. Description about auxins. Actually, Sachs reported that some growth promoting substance formed in leaf migrated to downwards. These Sachs experiments were confirmed by Charles Darwin and his son Francis Darwin by conducting phototropic curvature experiments. Phototropic curvature experiments. How means? They exposed coleoptile to sunlight. Exposed coleoptile to sunlight, then they noticed this coleoptile bent towards source of light. That is called phototropic curvature. So, phototropic curvature in a canary grass. Canary grass first reported by this Charles Darwin and his son Francis Darwin. Then they noticed their experiments in power of movements in plants. So, author of Power of Movements in Plants, this Charles Darwin and his son Francis Darwin. Then next scientist, Botany. This Botany, they placed gelatin between stump and coleoptile tip. Even afterwards also, this coleoptile tip shows bend. So, by this experiment, he reported that these growth regulators are a diffusible substance. And actually, credit for the discovery of auxins goes to Went. Went conducted experiments in oats. Scientific name of oats, 𝗪𝗚 sativa. So, point is, first, Went, he cut coleoptile tip. He cut coleoptile tip and placed that coleoptile tip on agar block. Agar block means agar agar. It is obtained from agarophytes like red alga, gracilaria and gelidium. After some time, he discarded coleoptile, then he placed agar block. He discarded coleoptile, he placed agar block eccentrically. Eccentrically means halfway on a decapitated stump. Eccentrically or halfway on decapitated stump. Then after some time, he observed, he noticed that a coleoptile bend. Why this coleoptile bend means? Actually, these growth regulators synthesized in coleoptile tip, when it is placed on this agar block, from tip, these chemicals migrated to agar block. When agar block placed halfway on this decapitated stump, only one side, these growth regulators migrated, then other side. So, only one side more growth takes place, and finally this coleoptile tip bend. So, rightly it is called 𝗪𝗚 curvature test. So, this 𝗪𝗚 curvature test conducted by Went, and it is rightly called bioassay test. So, bioassay test for auxins, that 𝗪𝗚 growth test and 𝗪𝗚 curvature test. Then actually auxin synthesized in a shoot apex. Auxin synthesized in shoot apex, little root apex. From shoot apex, auxins migrated downwards. It is called 𝗂𝗍𝗍 petalis. And some auxins also migrated upwards, called acropetalis. But dominant is 𝗂𝗍𝗍 petalis. This movement is 3:1. So, they move from one end to other end. So, rightly it is called that polar movements. Polar, these are called polar movements. Auxins, natural auxins. Natural auxins, indole 3-acetic acid, 4-chloro-indole acetic acid, 4-chloro-indole acetic acid, phenyl acetic acid. These are natural auxins. Then artificial auxins. Artificial, one is naphthalene acetic acid, indole butyric acid. Rightly indole butyric acid, both natural and also artificial. Then 2,4-D. 2,4-D means 2,4-dichlorophenoxyacetic acid. Then 2,4 5T means 2,4-trichlorophenoxyestic acid. These are natural and artificial. Most common natural oxygen is indolestic acid. This indolestic acid is produced from precursor for indolestic acid is amino acid tryptophan. Then zinc. Zinc is essential for this tryptophan. These are importance of oxygens. Now physiological effects of oxygens. One is cell elongation, then apical dominance, then tropical movements, then root initiation, parthenocarpy, and sex determination. So cell elongation, apical dominance, tropical movements, root initiation, parthenocarpy, and sex determination. So first one, cell elongation. So what is the function of oxygens means? Along with cytokinins, they promotes cell elongation either in coleoptiles or in artificial media like callus formation. The next one, apical dominance. Apical dominance means under the influence of apical bud. Under the influence of apical bud, these lateral buds become inactive, or by the influence of apical bud, that the growth of lateral bud arrested. That is called apical dominance. This was reported by 𝗬𝗏 and 𝗬𝗃. 𝗬𝗏 and 𝗬𝗃. So point is reported by 𝗬𝗏 and 𝗬𝗃. How this apical dominance can be removed means simple. Removal of this apical bud. Rightly, this removal of apical bud is called topping. Removal of apical bud is called topping, and topping results in the formation of lateral branches. Actually, if apical bud is more active, this oxygen synthesized migrated to downwards, and this oxygen stored in lateral bud. If more oxygen is present in lateral bud, they stop that growth. That is why these lateral buds remain dormant. If this apical bud is removed, then oxygen synthesis gets stopped. So in lateral buds, that oxygen concentration also decreased. So lateral bud begins to grow and gives branches. This process can be seen in that coniferous plants like a pinus, even casuarina. So it is cell elongation, apical dominance. Next one, tropical movements. Here, oxygens are responsible for tropical movements. It is pea seedling. Pea seedling, horizontally placed pea seedling exposed to sunlight, photo bleaching takes place or photooxidation takes place. So more oxygens synthesized at the base of root and also at the base of shoot compared to this illuminated side. Upper side is illuminated. Because of photobleaching, compared to illuminated, more oxygen synthesized in basal side. So presence of more oxygens in shoot promotes growth. So shoot apex bend upwards. In that way, shoot shows positive phototropism. But presence of more oxygens inhibits growth in root. So lower side more oxygens present, they inhibits growth. Upper side less oxygens present, they promotes growth. So root bend downwards. These are called tropical movements. These tropical movements were reported by 𝗂𝗽𝗍 and Went. So it is called 𝗂𝗽𝗍-Went theory. These are tropical movements. Next one, root initiations. Actually, oxygens, it is hard. Because of this reason, oxygens used in that horticulture, particularly in horticulture, lifespan of plant depends on formation of adventitious roots. So when some stem cuttings are immersed in this oxygen solution, so they produces roots. Most commonly, which oxygens are used means indole-acetic acid and naphthalene-acetic acid. But highly effective oxygen for the formation of adventitious roots, IBA, that is why this IBA is called rooting hormone. It is root initiation. Next one, parthenocarpy. Parthenocarpy means formation of fruit without fertilization. It's called a parthenocarpy. Generally, parthenocarpic fruits are seedless. That is why it is advantageous. So, ability of oxygens to induce parthenocarpy first demonstrated in that orchids. The next one, sex determination, particularly in cucurbids. Particularly in cucurbids, this oxygens increases number of female flowers. So number of female flowers in cucurbids increased by both oxygens and also that ethylene. And last one among oxygens, very important, most oxygens commonly used as weedicides or herbicides. Dicot weedicide 2,4-D. So dicot weeds can be killed by that 2,4-D. Some monocot weedicide. Monocot weedicide is dalapan. Dalapan means 2,2-dichloropropiononic acid. 2,2-dichloropropiononic acid. So these are physiological effects of oxygens. Weedicide, elongation, dominance, tropical movements, root initiation, parthenocarpy, and sex determination. Second type of growth regulator, gibberellins. Japanese farmers, they found that infected rice plant become thin, tall, pale, and colorless. For that disease, they gave the term foolish seedling or Botany disease. Foolish seedling or a Botany disease. Then reported that this disease is due to something secreted by fungus. That fungus is Fusarium moniliforme. Fusarium moniliforme. It is asexual stage. So, belonging to 𝗳𝗏𝗧𝗧, and perfect stage of this fungi is 𝗂𝗶𝗍𝗁 𝗧𝗕𝗓 𝗂𝗽, belonging to class Ascomycetes. Then sterile filtrates. Sterile filtrates of this fungus isolated by Savada and transmitted into healthy plants. Then he noticed that healthy plants got disease. Finally, 𝗹𝗶 and 𝗬𝗧𝗧. 𝗹𝗶 and 𝗬𝗧𝗧, they isolated this chemical in a crystalline form. For that, they gave the term gibberellic acid. And all gibberellic acids, they contains gibbon rings. And most of the gibberellic acids obtained from fungus. And first discovered and well studied gibberellic acid is Ga3. First discovered well studied gibberellic acid is that Ga3. It is non-nitrogenous containing phytohormone, and it does not show any polar movement. And auxins, they are nitrogen containing phytohormone. They shows polar movements. Shows polar movements. So, physiological effects of gibberellins. One is removal of genetic dwarfism. Then parthenocarpy. Then seed germination. Seed germination. Then bolting. These are major things. So, if these plants were treated with the gibberellins, these plants grow into normal height only up to that generation. Then next one, parthenocarpy. Like auxins, even gibberellins promotes parthenocarpy in grapes and tomato. Particularly in grapes, gibberellins promotes the formation of number of fruit bunches. Number of fruit bunches per plant, and also number of fruits per bunch, and also responsible for elongation of internode. Due to elongation of internode, there is wide gap between the fruits. So, if there is wide gap, they are less susceptible to diseases. And one more importance of gibberellins, they promotes seed germination. This was first reported by Palleg in barley. Actually, barley seed. In barley seed, there is embryo, This embryo is surrounded by endosperm. Embryo is surrounded by endosperm, but this endosperm is a starchy. So, this embryo can't absorb that starch. That is why at the time of seed germination, this embryo produce gibberellins, and these gibberellins, they enter into endosperm and they produce alpha amylase enzyme. This alpha amylase enzyme hydrolyze that starch. Alpha amylase enzyme hydrolyze that starch into sugars, then embryo absorb that sugars. Finally, seed germination takes place. Then there are some plants like cabbage and cauliflower. This cabbage and cauliflower, during vegetative stage, these plants become dwarf. These plants become dwarf during vegetative stage because that gibberellins are absent. Then just before flowering, gibberellins are formed. So, rapid elongation of stem takes place. This rapid elongation of stem is followed by flowering, is called bolting. Then next one, gibberellins increases number of male flowers in that cucurbita. Then it is used in brewing industry and also increases actually sugarcane that stem store that sucrose. Then gibberellins increases elongation of internodes. So, that amount of sugars increased. And gibberellins also promotes flowering in rose, rhododendron and poinsettia. Rose, rhododendron and poinsettia. Next one, bioassay test for gibberellins. Bioassay test means employment of living material. Employment of living material to test biological activities. It's called bioassay. Bioassay test for gibberellins. One is 𝗳𝗏𝗪𝗔𝗬 test and second one barley endosperm test. Barley endosperm test. So these are physiological effects of gibberellins. Removal of genetic dwarfism, parthenocarpy, seed germination, bolting, increase in male flowers, brewing industry and sugarcane content, even in flowering in rose, rhododendron and poinsettia.